Okay, so today we're going to be covering Chapter 10, host microbe interactions and pathogenesis. Okay. So let us go ahead and take a look at what we're going to be covering today. I'm going to go over the learning objectives, okay, which remind us of what we're trying to get out of this chapter. Okay. I'll open it up to questions, then we'll go through the steps of infection, virulence factors, safety in healthcare, and then what to do for next time. All right. So this chapter helps us move toward being able to meet the following learning objectives. Define the key role of evolution as it applies to microbiology. Identify microbial structures and connect the structures to their functions. Identify necessary components and processes in microbial metabolic pathways. Identify key agents and processes of microbial information transfer. Identify pivotal components of microbial systems important to health. We're going to be doing an awful lot of that today. And analyze and describe the impact of microorganisms. We'll be doing a lot of this one too. Whoops, I went too far. All right, any questions? Yes, Sherry.
So I know I read that the exam will be over chapters 1 through 8. Is there going to be some of 9 and 10 on there as well?
For chapter, for exam 3?
Yeah.
No, it stops at 8.
Okay.
It's mainly going to be over chapter 7 and 8. And remember, we split chapter 7 and 2 because I have a thing about infection control. I did that for many years. So, yeah, but you need to know chapters 1 through 6 to be able to understand 7 and 8. And then when we get to exam 4, then it'll mainly be over 9, 10, and 11.
Okay, thank you.
Quite welcome. Okay, any other questions? Oh, by the way, and I'll mention this at the end of lecture, but I may as well bring it up now. I was thinking about moving the date for exam 4, but then it starts running into exam 5. So what I did instead is I pushed back the due date for the signature assignment case study. It's not due next week. It's due in about 3 weeks. And so that'll give you time to study for exam 4 and not have to worry about getting that in right away. Unless you want to turn it in early, feel free to turn it in early.
Yeah, right. Thank you.
Oh, you're quite welcome. I understand completely. I do things in the order they are due, so I understand. Okay, any other questions? Okay, if you think of any, let me know. As you know, I'm happy to answer them. Okay, so the first thing that we are going to do is we're going to cover the steps of infection. So let's go to the whiteboard. By the way, if you downloaded the PowerPoint before about 30 minutes ago, I had virulence factors first, but in going through and reviewing what I was going to talk about today, that didn't make a whole lot of sense. So we're going to do the steps of infection first. Okay. Now, with the steps of infection, we're going to take this from the pathogen's point of view. Okay. So thinking at it, or thinking of it, okay, you can also think of this as goals of the pathogen. In later chapters, we're going to talk about this whole process from the point of view of our cells, our immune system. Okay, so how are we going to fight this pathogen? But for this part, we're going to talk about what the goals of the pathogen are. Okay, all right, so let's go ahead and get our for a host here. Okay, they're happy because they're not sick yet. Okay, and then we have the evil pathogen. Okay, we're just going to randomly put them floating out here. Okay, so what is the first goal? What's the first thing that this nasty evil pathogen has to do?
Attach.
Yes, actually, yes, it does need to attach, but we're going to put that second.
Okay.
They need to find the host first.
Yeah, they do need to find the host. Okay.
Is that
So-
attach?
Find is kind of combined in the first two.
Find a weekend host. Find a spot to get in.
There we go. Find a spot to get in. Okay. Okay. So they've got to find what we call a portal of entry. And it all depends upon the pathogen. Okay. So we humans have all sorts of portal of entries. Okay. We'll say this one is COVID. Where is it wanting to get in?
Your respiratory system.
Yeah, your respiratory system. So it can come in through the nose, which I haven't drawn, or the mouth, or the eyes, because the eyes are connected to the respiratory system through the sinuses. Okay. And what if we are talking about a waterborne pathogen? Okay, we'll put the waterborne pathogen in blue because it's carried by water. How does it want to get in? Depends upon the pathogen, right? We'll say this one causes vomiting and diarrhea.
The mouth.
The mouth. Very good. Okay. Okay, so different pathogens have different portals of entry. And it all depends upon where they want to attach. Okay. Because attachment is important. Okay. Because, and we'll talk about this in virulence factors. Oh, well, let's talk about it now. Let's combine virulence factors because they are an important part of the steps in infection. Okay. Okay. Virulence factors are structures or products of a pathogen that can cause disease. Okay. So the more virulence factors a pathogen has, the more severe disease it causes. Okay. Virulence is a measure of the severity of disease. Okay. So if I have a microbe that is avirulent, okay, it doesn't have any virulence factors and it doesn't cause disease. Okay. Now that's an oversimplification. Okay. But if you have, an opportunistic pathogen, okay, it has some virulence factors, okay, but not too many of them. And then you have, true pathogens, okay, and they tend to cause severe disease, and they're generally the ones that we're most worried about. Okay. Now, like I said, this is an oversimplification, and we'll talk about the various virulence factors and whether they contribute to avirulent, opportunistic, or true pathogens as we go along. Okay. All right. So attachment. There are structures on pathogens that allow them to grab a hold, okay, to attach. So I know this is going to be a stretch, but let's talk about bacteria first, okay? Let's say that this waterborne pathogen is a virulent strain of E. coli, okay? So what structures, thinking back to chapter 3, I know that's a long time ago, what structures are going to allow it to attach?
The spikes.
You know what? Let's go ahead and put spikes down, but I'm gonna put that down for viruses. Very good. So for my COVID here, the spikes are definitely going to allow it to attach, and that's considered a virulence factor. Okay.
Flagellum? Flagellum?
Flagellum are virulence factors, but they're more involved with the portals of entry. They allow them to swim and move around. So let's go ahead and put flagellum up here, because, you know, my E. coli, when they are swimming around in the water, they've got to have something to swim with. Okay.
Would it be glycolics?
Oh, a glycocalyx? Yes, yes. Some glycocalyxes, okay, allow a bacterium to attach to another cell. Okay. So we're going to put glycocalyx. Okay. And they come in two flavors. Okay. They can either be a slime layer, or they can be a capsule. And capsules are going to show up again, okay? Because depending upon the pathogen, capsules have different jobs, abilities, okay? But in this situation, you know, a capsule can attach to another cell, okay? Say along your intestinal tract, or if it's a respiratory bacterium in your sinuses, okay? Well, what's another one that also starts with an F, like flagellum?
Flagella.
But yes, but what's a different structure?
Is it philia or phila?
Fimbriae?
Fimbriae.
Very good. Yes, fimbriae. Okay, so on my cute little bacterium, okay, fimbriae are these little hairy looking things that you would think were cilia, but they have a different structure. Okay? So that would be your fimbriae. Okay? And if I had a capsule, then a capsule would be kind of like here. Sometimes the fimbriae stick out from the capsule. Okay? So yes, these are structures that are involved in attachment. And let's go ahead and get our COVID up here. Since unfortunately we've been having to deal with COVID for the past 2 years and probably for the rest of our lives. That's kind of a depressing thought, isn't it? But oh well. And because it's an envelope virus, we'll go ahead and put an envelope on there. Okay. All right, so these are structures, okay, that enable the pathogen to grab a hold, okay? And they are all very, very specific, okay? And that's why finding the right pathogen has to do with the portal of entry, the spot to get in, as well as attachment, okay? Because, you know, E. coli really doesn't want to attach in your sinuses, okay? If it gets into your nose, it's not going to grab a hold until it gets down into your intestines. Okay? These fimbriae are very, very specific to what kind of human cells they want to grab a hold of. They're not going to grab a hold of the stomach, okay? Because they're just trying to get through the stomach as fast as they can, because they are not acidophilic. They're not going to grow in your stomach, okay? Same thing with viruses. Viruses are very specific as to where they want to attach. Okay. So even though we have found COVID viruses in wastewater treatment, okay, in feces, there's some indication that they might replicate in your intestinal cells and cause some vomiting and some diarrhea. Okay. They are unusual. Okay, most viruses are very, very specific in where they want to grab a hold. Okay, so to use an example, okay, herpes simplex virus. Okay, there's type 1 and there's type 2. Type 1 is generally associated with the oral mucosa, okay, the oral mucous membrane, your lips, the inside of your mouth. And then herpes simplex type 2 is generally associated with the reproductive tract, okay, vagina, cervix, anus, penis, that kind of thing. And it's because their spikes are specific for the cells in those areas of the body. Okay, now they can get in other areas, but they really prefer those areas. Okay, and it's kind of like COVID really prefers the respiratory tract. Okay, all right. Any questions about attachment? Okay, and I got all excited about attachments. I lost my train of thought, so I had to look over there. All right, so what's the next thing, okay, after it attaches, okay? There's a couple of things the pathogen has to do. And the book leaves one out. I don't think I'm going to test you on it, but I'm going to mention it.
Is this where it penetrates, or is that also something different?
No, you are on the right track, because if we're talking viruses, penetration is the next one. Okay.
Okay.
But we're going to generalize this. So I'm going to go ahead and put penetration.
Would that be uncoding?
Yep, penetration and uncoating. Now these are specific to viruses, but we're going to tie that into what bacteria do too. Okay. So this is a part, okay, of the general step of invasion. Okay. So if we're talking the virus, penetration and uncoating is where it invades the cell. Okay, it gets inside of the cell. Okay. Whereas if we're talking about E. coli, and we'll say that this is hemorrhagic E. coli, okay, 0157H7, I think. Then I lost it. But anyway, hemorrhagic E. coli. It needs to get past your intestinal lining into your bloodstream, okay, to cause, okay, bloody diarrhea, which is dysentery. Do you ever hear about dysentery? Yeah, that's what they're talking about. It's bloody diarrhea. Yay, right? So it penetrates, gets into, invades the lining of your intestines. Okay, and of course, that's going to cause bleeding. Okay. And if the patient is really unlucky, it's going to get past there into the general circulation and find a home in your kidneys and start shutting down your kidneys. Okay. So all of that would be involved in invasion. Okay. Now, like I said, penetration and uncoating is what is the viral version of invasion. Okay. We're talking bacteria. Sometimes they do the same thing. There are some bacteria that will only grow inside of our cells. Okay. But there are some like E. coli that just want to get into your tissues. They want to get into your tissues because there's all sorts of deliciousness in there. Okay. So what would help a pathogen invade? Let's go ahead and start with viruses. What structures on a virus are going to help it get into the cell, invade the cell through the process of penetration and uncoating?
The spike.
Spikes again?
Spikes again. Yep, very good. Spikes are kind of dual purpose. Okay. They grab a hold. Okay. And in animal viruses, they help the virus get into the cell. Right. Now, if we're talking bacterial phages, you know, part of the capsid helps do that. But for this chapter, we're going to stick to animal viruses. Okay. All right, so they're important in invasion. Okay. Now, for this part, we haven't talked about this kind of thing in a long time, but for bacteria, okay, they can produce toxins. Toxins are generally small proteins, peptides, okay, that do a variety of things. Okay, they can pop cell membranes, okay, and they can cause cells, okay, to bring them in, okay, so they can be a part of what intracellular pathogens use to get into the cells. They trick the our eukaryotic cells into thinking this is a delivery, kind of a Trojan horse kind of thing. Okay. And they can do all sorts of different things. Okay. Some toxins specifically target neurons. So we call them neurotoxins. Okay. Some of them specifically make you vomit. Okay. And we'll be talking about these enterotoxins. Okay, later when we talk about how the pathogens get out. Okay. Some of them specifically target white blood cells. Okay. So these are peptides that do all sorts of nasty things. Okay. But the ones that we're interested for invasion are ones that would kill a cell or trick it into taking the pathogen in. Okay. Now, there are also toxins that are enzymes. And they are enzymes that are released by the pathogen. Okay, so this is my coli, and I'm going to release an enzyme that we call an exoenzyme. Okay, that can digest things, can break things down. Okay, so that nutrients go in. Okay. So they can break down proteins, lipids, carbs, you name it. Okay. All of that would allow the pathogen to get into deeper tissues. Okay. Now, not all pathogens invade the tissues. Okay. All viruses go inside of the cells, but not all pathogens invade the tissue. Some of them hang out, grabbing a hold of our mucosa or our skin, and they release toxins that cause disease. Okay. But not all of them get in. And the book kind of leads that one off. But I feel I have to tell you about it. So that you understand what's going on. Okay.
Teresa, do they get in deeper because the enzymes have broken everything down and then they go in with the nutrients and get in deeper that way? Oh.
Yeah, nutrient acquisition and invasion are kind of together in that one. Yeah. But then some of them also, like, will release enzymes that will break down the proteins that are holding the cells of your tissues together and kind of open up a doorway without popping the cell. So enzymes that would break down the protein collagen, which is important in holding the cells of your tissues together, that would help with invasion. There's also another protein. Actually, it's not a protein. It's a carbohydrate. But anyway, there's another molecule called hyaluronic acid. If you use skin cream to prevent wrinkles and to moisturize your skin, you will have heard of hyaluronic acid. Okay? It helps hold your tissues together, and younger folks make more of it than older folks, which is why they put it in beauty creams. There are pathogens that will break that down, and that will also open up a doorway from your mucosa into your tissues. Okay. So, by the way, on a test, I would not expect you to know these different types of exoenzymes and toxins But I would want you to know, if I describe what they do, I would want you to know whether they are important in invasion or nutrient acquisition or attachment or, yeah, does that make sense? What I'm wanting you to get from this? Okay, so the more virulence factors, okay, a pathogen has to get deeper into your tissues, the more severe disease it causes, okay? So, you know, the E. coli that's very happily living in my colon and not giving me bloody diarrhea, okay, do not have the toxins or the exoenzymes to pop my cells, okay, or to eat their way into, you know, the past the lining of my intestines, okay. So they would be considered avirulent, okay, or very low virulent. Whereas hemorrhagic E. coli, because it has all of these virulence factors, is considered very virulent, okay. Are we getting a feel for what virulence means? Okay, so we already brought up another goal. One of the reasons why a pathogen wants to get in is to get more food, and we call that what?
Essential nutrients.
They are essential nutrients. And so nutrients are in there. Nutrient. And we're going to use a fancy dancy word for getting. Okay, nutrient acquisition. And I can't even spell it. There we go, that's close. Okay, so it's a fancy dancy term for getting food. Okay. Now, when we're talking viruses, technically, viruses do not do this, okay? Because they're not alive. And so if they're not alive, they don't need food. But they do get into the cells and they do take over, but we don't call this nutrient acquisition, okay? It's getting in and it's taking over the factory to make something different, okay? I know that's kind of a weird concept because we talk about viruses eating cells. They don't really. Yeah. The phage meaning, you know, Greek for eating. So watch out for that on the test. If it's a virus, there is no nutrient acquisition, okay? But bacteria, protozoa, okay? Fungi, anything that's alive, okay, is going to be interested in getting food, okay? So as a tie-in to the first part of chapter 7, okay, pathogens generally do not have a lot of metabolic pathways to make their own stuff. They need a lot of vitamins, okay? They can't photosynthesize. Yeah, that's an exaggeration. There are some poisonous plants, but they don't infect you. They create toxins to keep you from eating them. It's more of a defensive mechanism, okay? But so far as I know, there are no algae and no plants that will grow inside of you and cause disease. Thank goodness. That's a horrible thought. Okay, so these guys are after products they can't make themselves, okay? Vitamins, okay? Minerals they can't get from the environment because they're not plants, okay? And they're also after a carbon source. So all pathogens that will grow inside of a human or on a human are chemoheterotrophs. Yay, right? They need to eat us. And so they have certain ways of getting their food, to get their nutrients, to get their essential nutrients, okay? Some of them are these toxins, okay? Especially the exoenzymes and the toxins that will pop cells, okay, and digest proteins, lipids, carbohydrates. Okay. So these kind of do double duty. They can help them get into deeper tissues and they can also feed the pathogen. Okay. There's one other one that I want to mention called cyanophores. Okay. Cyanophores are all about getting iron. Okay. We need iron. We need iron in our hemoglobin to carry oxygen. Okay. We need iron for some of our enzymes. It's a cofactor in, cofactor in some enzymes. Okay. The enzyme isn't going to work unless we have a little bit of iron. Okay. Trace nutrients. Well, because iron and magnesium and some of these other minerals are so important to us, and there's just a little bit of it, we have proteins that carry them. Okay. So using iron as an example, okay, if I'm going to move iron around my blood, I use a molecule called transferrin. And by the way, ferrin is Latin for iron. So whenever you see ferrous or ferrin, you know they're talking about iron. Okay. If I'm sending iron to my little baby in breast milk, okay, that would be lactoferrin. Okay. Well, some pathogens produce these molecules called cyanophores that have a greater attraction for iron than our molecules. Okay, so basically these cyanophores will steal, whoops, that's supposed to be an F, okay, will steal iron from our carrier molecules, our carrier proteins. Okay. And that's considered a virulence factor that is related to nutrient acquisition. Okay. Meanwhile, while they're in here invading and getting food from us, our immune system responds. Okay. Da, 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 da. Okay, great big white blood cell. White blood cell comes in and it's going to phagocytize these guys once it realizes that they're there. Okay. So one of the jobs of a pathogen is to hide. Okay. Hide from the immune system. So we call this immune system evasion. And I have officially run out of room on my whiteboard. Are we okay if I clear the whiteboard and kind of start over? Okay, so I'm going to put the steps that we have so far. Okay, we have entry. We have attachment. We have invasion. We have nutrient acquisition. Okay. And now we're talking about evasion of the immune system. Okay. My apologies that evasion and invasion sound similar, but at least they start with a different vowel. Okay, so I'm going to get my bacterium and my virus set up here. Okay, as we talk about how they hide from the immune system. Okay. And let's go ahead and get some fimbriae in here because that becomes important. Have them stick outside of my capsule here. Okay, and so the body has realized that there's a problem. Okay, whenever cells get popped, they release a chemical signal saying, help, help, help. Okay, and I'm going to put my big old white blood cell here. Okay, so the goal of the white blood cell is to eat these guys down. Okay, but the pathogen's goal is to hide. Okay, you got to hide from the other team. So they do a number of things, okay, that we group under mimicry. I think I spelled that right. Just had a brain freeze. Masking and variation. All right, so I'm going to introduce a term that we're going to be talking about in chapter 11 too, okay? And it's called antigen. Okay, antigens are proteins, generally speaking, that stimulate an immune response. Okay, so it's anything that your immune system sees as foreign. Okay, so the spikes on the COVID virus, okay, would be an antigen. Okay, the fimbriae on E. coli would be an antigen. Okay, the capsule, okay, sometimes there's some protein sticking out, that would be considered an antigen. Okay, flagella, okay, are considered an antigen. Peptidoglycan is an antigen. Okay, anything that is foreign, okay, is going to stimulate an immune response. Okay, so all of these, and by the way, we abbreviate this as AG. Okay, so we have antigen mimicry, antigen masking, and antigen variation. Okay, so let's talk about variation first, because we're most familiar with that with COVID. Okay, over time, okay, the antigens on the SARS-CoV virus have changed. Okay, originally, okay, they were alpha, and then they changed to what we call
Delta?
Delta. And then which one are we dealing with currently?
Omicron?
Omicron. Okay, and there's some other ones showing up. Okay, we call these strains. Okay, because through random mutation of the RNA genome, because RNA polymerase is not very good, okay, and so we get mutations, we get slight changes in these spikes. Okay, and so the antibodies that the body produces, okay, either through vaccination or from getting the virus, okay, that are intended to cover up the spikes and prevent attachment, okay, don't work as well because, you know, we get different shapes. Okay, that's variation. Okay, now in a virus, because it's not alive, okay, this is from random mutations. Okay, so mutations can lead to antigen variation. Okay, and in previous chapters, remember there's small variations, slight mutations, spontaneous mutations that lead to, whoops, drift. Let's get them right. And then if you get big changes, then we call that shift. Okay, and bacteria do this too. Okay, they have a DNA genome, and so DNA polymerase is better at what it does. Okay, and so they mutate more slowly, but they do change over time. Okay, all living things, the populations can change over time. Okay. Now sometimes they do it on purpose. So like with my cute little E. coli, with its genome, when it's out swimming in the water, it produces a flagellum. And it doesn't make fimbriae. But once it gets into your body, it says, oh yeah, I want to stay here for a while. It will stop making fimbriae and stop making flagella, and it will start making fimbriae. And so that is antigenic variation. Because if your body has antibodies against the flagella, they're not going to work anymore. It's got to start recognizing these fimbriae and make antibodies for those. Okay. So some pathogens do it on purpose. Well, on purpose, they switch which genes they are using. They are transcribing and translating. Okay. And others, it's mutations. Okay, random, spontaneous mutations that cause a change in the antigens. Okay. How are we doing? See how this all ties into stuff we talked about previously? Okay, masking. Okay. Sometimes they hide their antigens. Okay. So for example, this is an example of where the capsule can be important in an evasion. Okay. Because, oh yes, Michelle.
I want to go back to what you said about transcription and translation in the variation.
Mm-hmm.
So when it gets in and it drops the flagella and gets the fimbriae, that's part of what's going on in transcription and translation, right?
Right.
And that is something that has evolved over time. Like it got inside and went and died and decided, hey, I want to stay in here. How can I stay in here? Or was there a mutation that happened that let it do that?
So good question. Okay. So E. coli have evolved over time to be able to turn on and turn off genes. Okay. Because if you're not swimming around in water, you don't need to make flagella. But when you're out in water, you do want to make flagella so that you can swim around, okay, looking for a new host. I say looking, they don't have eyes. Okay, so we'll say that there's a gene right here, okay, that makes flagella. It codes for flagella. And so when it's turned on, it gets transcribed into RNA, okay, and the ribosomes grab a hold of that and make flagella proteins, okay, and then these proteins get moved over, and because E. coli are paratrichous, okay, they have multiple flagella, okay. So once they get inside of a body, they say, oh, we don't need the flagella anymore. We're going to turn this gene off, and we're going to turn on the fimbriae gene, okay? So it's going to stop making flagella proteins, and it's going to get rid of its flagella. It's going to snip them off, and it's going to start making fimbriae proteins, okay? So we're going to get these short little fimbriae proteins that are going to get moved to the outside, and that's going to enable the bacterium to grab a hold of intestinal cells. Okay. So here's a human example, okay, that will make it easier, okay, to understand, okay. Let me think of a good human example that turns on and off. Oh, okay. So, if I am a salivary gland, okay, in my cheeks, okay, I have genes turned on to make the digestive enzyme amylase. Okay, it breaks down starches, like if I eat a cracker, it's going to break it down to glucose. Okay. My brain cells have the same genes that my salivary glands have, but they don't produce amylase. There would be no reason for them to produce amylase. So those genes are turned off. They have the genes, but they don't make them. Okay. My salivary glands have all the genes to make neurotransmitters, but it doesn't make sense for them to make them because they're not neurons. So those genes are turned off. Okay. So E. coli do a similar thing. They have genes that they turn on and off as needed. Okay, does that help?
Okay, and they evolved into that?
Yes, over time, E. coli developed the ability to do that.
Okay,
Yes.
okay. Thank you.
Oh, you're quite welcome.
So that being so extreme, then Teresa, would that be a drift variation?
Oh, turning genes on and off? No,
Shift?
that would be a variation that is called phenotype. And that's what the genes they're currently making. Yeah, does that help?
I guess I'm still wondering, and I know it's not maybe part of the test. So I'm sorry if I'm wasting anybody's time, but
Yeah.
how did it learn that it needed to turn it off and it needed to turn it on? You know what I mean?
Yes, that is a very good question. Okay. And the answer is it's kind of related to smell. They have little antenna, kind of like smell. It's kind of like they have little scent receptors all over them, or you can think of them as little antennas. Okay. And that tell the inside of the cell what's going on the outside, so that it can feel or smell if it's in open water, and it can feel or smell if it's inside your body. And based on that, it turns on and off different genes based on these receptors on the outside of the cell.
And they go in, they entry, attach, invade. Is that all for the purpose of nutrient acquisition? And then once they get in there, they evade so they can stay in there?
Yes.
So this is just them wanting to live and replicate and get bigger. And some of them are causing us problems.
Exactly.
Okay.
Because of these virulence factors that cause us problems. Yep.
That go along with them. Okay. So if it didn't have any virulence factors, then it would be like, who cares?
Exactly. Your immune system would leave it alone.
Okay.
Yeah. So, they all have attachments. Your friendly E. coli in your gut have to grab a hold of your intestinal cells. Otherwise, they can't stay there and make vitamin K for you. The problem occurs when they get these virulence factors that allow them to invade and start popping your cells.
Okay, thank you.
Yeah, the E. coli that are in our gut that are not causing a problem, they have to wait until we eat to get food.
Oh.
They have to be patient.
Okay.
Pathogens are not patient. It's like, I want food now. You don't feed me, I'm going to start killing your intestinal cells. Kind of like, the Godfather, you pay me the money or I break your knees.
Okay, I got it. Thank you.
Does that help? Yeah. So, speaking of the Godfather, so some of this masking is hiding. Okay, so it's kind of like back before COVID. If you went into a store wearing a mask, we knew you were up to no good. But it was hard to catch the criminal because we couldn't tell from the mask, who they were from the video surveillance, okay? So some pathogens do that. They cover up their antigens, okay? And that's part of what a capsule does. Okay, capsules are made out of carbohydrates, and carbohydrates are bad antigens. They generally do not stimulate an immune response. It's like your white blood cells can't see a capsule if it's mainly composed of sugars, okay? And they're also kind of slippery. It makes it hard for this white blood cell to grab a hold. So if it does grab a hold of it, it kind of, is slippery and it slides away and it has a hard time grabbing a hold, okay? So a capsule can be involved in masking, okay, and hiding. Now with our virus, let's say that this white blood cell is infected with a virus, okay? It's doing all of its good stuff in here, all of its synthesis and assembly, okay? And so other white blood cells can't see them, okay? Until, if they're an enveloped virus, they start putting their spikes on the surface. Okay, that's the first time your immune system can see them, okay? Because they're masked, they're hiding, okay? So if it lives intracellular, that is a way of evading the immune system through masking. It's covering itself up as a cell, a healthy cell, nothing to see here. Huh? Now, mimicry is where it gets kind of scary. That's where they produce antigens that are the same as yours. Okay, they look like human antigens. Okay, so there are some pathogens that cover themselves in hyaluronic acid. So they look like they are the molecules that are connecting the cells together. Okay, so your immune system, goes through, takes a look at it, and it says, oh no, this is part of the human body. It's supposed to be here. Scary, huh? There are some viruses, and we'll get into this when we talk about the immune system, that make an immune protein, okay, that they display on the surfaces of the cell, okay, because otherwise, when they get in there, they're going to stop the cell from making their own proteins, okay? And so they would normally stop making this MHC, major histocompatibility complex. So what they do is they bring their own, and it looks enough like the real thing that an immune cell will go along and say, oh yeah, you're fine, there's no problem inside. Yeah, so mimicry generally involves making proteins that look like human proteins. And it takes your immune system longer to figure out what's going on. Kind of scary, isn't it? And then lastly, this isn't included in these three, okay? But there are toxins that target the immune system, okay? There are toxins that will specifically kill white blood cells. We call them leukotoxins, okay? And if you shut down the immune system, you're evading it, okay? And then HIV preferentially grows in white blood cells. So that's evading the immune system. If you're eating the immune system, you're not getting knocked down by it. Okay, how are we doing so far? Am I flipping your brains inside out? So for a lot of this, we've taken what we learned in previous chapters, now we're adding to it. Okay. Let's see if I'm missing anything. Yep. we only have one more step in the infection, okay? And that is exit. Okay. Okay, so eventually, no matter how good you hide, okay, it's going to be time to move on to a new host. Okay, we get in, we pillage, we leave. Okay, so we've got to get out. Okay, and so we call these portals of exit. Okay, so if I am a respiratory pathogen, how am I going to get out?
A sneeze.
A sneeze. Yes. We're going to go out the same way we came in, just more explosively. Okay. If I am a digestive system pathogen, okay, how am I going to get out?
Defecate.
Defecate. And what if I'm coming back up?
Oh,
Throwing up.
Yeah,
Vomit.
throwing up. Yep. So here's what pathogens do. Okay. If I am a respiratory pathogen, okay, the immune response, okay, irritates your respiratory tract, which leads to coughing and sneezing, extra mucus production. Okay. That's a part of the immune response, but it also is how the pathogen gets out. Okay. If it didn't irritate your system enough to cause you to cough or sneeze, COVID would have a harder time getting out to the next person. Okay. Now there are toxins that help digestive pathogens get out. Okay. So I'm going to go ahead and put immune system irritation. Okay, the immune system and trying to clear things out irritates things. Okay, and it helps the pathogen get out. That's not the intent of the immune system, but the pathogen takes advantage of that. Okay, toxins. Okay, there are some pathogens that produce toxins that cause diarrhea and vomiting. And the whole point is not invasion or nutrient acquisition, it's to get out. Okay, so toxins can do a whole bunch of different things. It just depends upon what they're doing as to which job they're fulfilling. Okay. All right, how are we doing? We doing all right? Are you ready for some questions? Questions to practice what you're learning. Okay. All right, let's talk about C. diff. Can you tell I have favorite pathogens? It's not that I would want to get them. They're just, oh, there we go, 5. Yes, I want 5 answer options. Okay. Okay. Clostridium, it used to be called, now it's called Clostridioides difficile. As a pathogen of the digestive system enters the body through which of the following methods? And yes, there is more than one right answer. We just passed 50%. Good job. Okay, we're at 76. See if we can get to 100. Okay, we're at 84. I need 2 more. There was 92. We're almost to 100. Come on, we can do it. We can do it. All right, for the sake of time, I'm going to shut it down now. Okay, the favorite answer is the GI mucosa. Correct answer. Tell me why.
Because that's how it gets to your digestive system.
Very good. Yes. So it enters through the mouth, it goes down, okay, and it grabs a hold. Okay. All right. The next favorite answer was respiratory. Probably not. Okay. Only if, say, you know, it's going from your sinuses down into your stomach. It is not the preferred portal of entry. Okay. So if you breathe in C. diff endospores, they're going to end up in your lungs. They're not going to be happy. They're going to die. Okay. 99% of the time. Okay. Ocular, yeah, maybe, you know, if you're cleaning up some C. diff containing feces, or if it's microscopic amounts and you don't see them and you rub your eyes, it can go from your sinuses down into your digestive tract. Not a real common portal of entry, though. Yes, Michelle.
I guess I'm getting, now that we went through the respiratory, how does it get in your GI mucosa if you're not like breathing it in or something? Is it because you're eating it?
It's because you're eating it unintentionally, of course.
Okay, so you have your respiratory mucosa, you have your GI mucosa, and they're infected by different ways, by breathing and by eating.
Yeah. Now, like I said, you can get it through your eyes into your sinuses. You can get it through your respiratory tract, but it's not as common. Because what it'd really rather do is go through your mouth and down your throat. As opposed to through your sinuses. You've got to get through all of your sinuses and hope you end up in the throat versus in your trachea, in your esophagus versus your trachea. It is just more fraught with danger for them.
Okay.
Does
Thank
that
you.
make sense? Yeah.
Yes.
And if a respiratory virus gets into your digestive tract, usually they're not adapted to survive the acid bath of your stomach. Whereas if it gets into your lungs, that's where it really wants to be. So these two are possibilities. They're just less likely. Okay. This one I would have accepted as a correct answer because we have seen some C. diff passed through anal sex. So it's a possibility. Not as likely, but it's a possibility. Okay. Okay, so this is the best answer. This is low probability, so probably not. It could. So it's a partially correct answer. Okay, how are we doing on this one? Okay. What if we were talking about SARS-CoV-2, which causes COVID? Now pick its favorite method of getting in. And there's more than one right answer. If this were on the test, I'd make it a multiple answer question. Ooh, we're at 70%. I was stalled at 84. How much time we got? Yeah, for the sake of time. Okay, favorite answer, respiratory. Good job. It's a respiratory virus. It really wants to get into your lungs, okay, or your sinuses. So mouth, nose. I would include ocular in that because you rub your eyes, it goes straight into your sinuses. It's kind of a direct route. Okay, so even though the virus doesn't infect your eyes, because the tear ducts are considered part of your eyes and it connects directly to your sinuses, I would consider both of those a correct answer. Okay. GI mucosa, I would say that's a correct answer because we find it replicating in your intestinal tract. Who does that? If you're a virus, you're usually not that generalized, but SARS-CoV is. Okay, so I would accept that as a correct answer. Okay, parenteal, that's where you get a break in your skin. Probably not. It really does not want to get into your deeper tissues. So if it were to get into a cut or to be accidentally introduced through, say, a hypodermic, you would probably be very, very, very unlikely to get it. Okay. So slight possibility because nothing is 100% in biology, but I would say no to that one. Okay. So if you pick parenteal, no worries, we're practicing. All right, let's go on to the next question. Okay. Let me relaunch the poll. Okay. All right. So the new vaccines, well, they're still new. Vaccines against COVID block which step of the infection? And there's more than one right answer. There's one that's a little more direct. Okay, we're past 50%. We're at 70%. Ooh, 76. Let's see if we can get to the 80. And I think we stalled at 76% for the sake of time. I'm going to end the poll. Okay, favorite answer, adherence to host tissues. Very good. Yes, the vaccines stimulate your immune system to produce antibodies, which cover up the spikes, which prevents it from attaching. The fancy term is adherence. Okay. The second favorite answer is D, evasion of host immune defenses. That is also true. Okay, because if you warn the immune system ahead of time, the virus can't evade the immune system. It's there checking. Woohoo! Okay. Also prevents evasion of tissues. Okay, so sure, I would pick that. Okay, and if you stop all of that, then yes, it prevents transmission to a new host. Okay, the only one I would say not so much is entering the host. Having antibodies does not prevent you from breathing it in. Okay, if somebody is coughing and sneezing in your general area and you breathe in the viruses, a vaccine is not going to stop that physical process. Okay, but you are on the right track. Okay, any questions? Questions about why I picked which ones? By the way, I like this one the best, but they're all correct answers. I would have to make a question like this on a test, a multiple answer question. Okay, let's go on to the next question. I think we have time for just one more. You're going good. Okay, cholera, the causative agent of cholera, Vibrio cholera, produces a toxin that causes large volumes of water to enter the intestines from the tissues. This results in profuse watery diarrhea. Which step of infection does this relate to? I'm going to relaunch the poll. And I'm going to say this time there's just one right answer. Okay, we're at 30%. We're almost to 50. Oh, we passed 50. See if we can begin. Oh, we're at 76. Okay, this one was a little harder. Okay, so the favorite answer was invasion of tissues. And you would think so, but the toxin does get into the cells, but the pathogen does not. So I know that's kind of splitting hairs. That's not my fault. That's how we define this kind of thing. So it's not really invasion of tissues, okay? Because the bacterium is not getting in, okay? Entering the host, you could say that, but we're going to say this is from the point of the pathogen that's already in the host, okay? Not so much, okay? Evasion of host immune defenses, not so much, okay? Because they're already there causing problems, okay? And later the immune system will clear it out if you give it enough time, okay? Adherence to host tissues, the exact opposite actually. Diarrhea is all about transmission to a new host. So I would say that that was the correct answer for this one, okay? Anytime we're talking about something that is causing diarrhea, vomiting, coughing, sneezing, phlegm production, okay, is all about getting to the next host, okay? Okay, any questions as to why this is the only correct answer?
Yeah, I have a question.
Sure.
Well, the question for this slide is, it causes it to enter the intestines from the tissues. So to me, that would right, but from the toxin that entered the body.
Right, right. So the toxin does enter the cells, but we don't consider that invasion by the pathogen itself. It's kind of like if it's releasing exoenzymes, and those exoenzymes are not causing the pathogen to be able to get into the tissues, we don't include that in invasion. Don't ask me why, but we don't.
So this step of infection is transmitting it to a new host.
Mm-hmm.
Would that be a new person then?
Yeah, a new person. All of that diarrhea, it's hard to keep it cleaned up, and eventually somebody's going to unintentionally ingest microscopic amounts. And microscopic amounts have large amounts of cholera. And it tends to get into the water supply, and you drink contaminated water. You don't know it's contaminated. It tastes fine, but you're ingesting large amounts of cholera. Yes.
Okay, so what would invasion of the tissues be for cholera?
Cholera does not invade the tissues. It sticks in the intestines. And that's why not all pathogens do the invasion of tissues.
Oh, okay. So not these 6 steps are not consistent with every pathogen.
Right.
And how do you know that?
Oh, I would put it in the question.
Okay.
Does that help?
Yes.
Yeah. Yeah, for the question, I want you to take a look at what I've described and see what it matches up to best. So for this one, it's getting out of the body in the form of diarrhea, and which would allow it to get to a new person, to a new host. So in retrospect, yeah, I probably should allow entering the host, because it would enter a new host. Maybe. Okay, other questions before we move on to what to do for next time? Okay, I will have some practice questions in the practice quiz so you can practice with some of these, and I'll have feedback so that you know why the correct answer is the correct answer. Okay, so let's move fast here. Okay. Oh, so we did not have time to cover precautions that we take in healthcare settings to prevent transmission. Okay, but most of it's pretty straightforward, whether we're using droplet transmission or droplet transmission preventative measures, or whether we're talking, universal things that you do for everybody, no matter. Okay, so make sure you read through that section in the textbook. Okay, exam 3 is open until Saturday. Okay, if you haven't taken it yet, go ahead and take that. Work on this signature assignment case study, but I pushed back the due date. It is no longer due next week. It is due 3 weeks from now. I pushed it back 2 weeks, something like that. Anyway, I have the new dates in Blackboard. Okay. And then next week, we're going to start on chapter 11, which is the start of the immune response. Okay. So we've talked about what the pathogens do. Now we're going to look at what we do to fight those pathogens. Okay. All right. Any questions?
Teresa, did you say that you weren't going to have office hours today? You were going to have them tomorrow instead at the
Right.
same time?
Actually, it's from 2 until 4, because I have a class from, that interferes with, say, a 1:30 to 3:30.
Okay, thank
So
you.
they'll just start like half an hour later. Yeah.
Great.
Okay, other questions? All right.